Hello everyone, and thank you for joining this tutorial video on how to use our data templates and to submit your data for the integrated report. I am Dan Brown, and I will be walking you through this tutorial today. Our first item is the agenda. What we'll go over today is why your data is important to us, the different data sources that we accept during this integrated report call for data. Then we'll go over some data requirements and talk about the submission window. And then we'll get into a live demo and then talk about how to submit your data. So getting on to our first point, this is why your data is so important to us. Here are the foundational steps of our integrated report. First are our water quality standards. These are criteria that protect the beneficial uses of our waters and can be numeric criteria values or narrative criteria values. The second step is data and information, and this is where your data comes in, and this is why your data is so important. This data is assembled by DEQ from publicly available data sources and the data you submit during the call for data. Without this, we would not be able to move on from this into the assessment methodology or creating the integrated report. Our assessment methodology explains how all the existing and readily available data and information is assembled and used. It's used to assess against numeric and narrative criteria. Our assessment methodology must be scientifically and technically defensible and must be revised each assessment cycle through a public process. It can require peer review and additional processes per state regulations. It does not require EPA approval. The integrated report, that final step, is how we, DEQ, report on the status of all state waters per the Clean Water Act, Section 305B. The second part of that is a list that we create of identifying the impaired waters or water quality limited waters around the state, and that's part of the Clean Water Act, Section 303D. Moving now into the data sources for the integrated report, we have three main sources. The first is DEQ-owned data, and this is data collected by DEQ employees or groups working with our volunteer monitoring program. Second is our readily available data. Much of the legwork here is already done, so a bulk of the work will be on DEQ to find the data that matches our data window and retrieve it from these databases. Readily available data pertains to any publicly available database that uses a common reporting structure and contain the requirements, the metadata requirements that we're looking for. If you know of a database that we should be aware of, please feel free to share connection information with us in an email after you finish watching this tutorial. The final data source is the call for data. During the call for data, we provide the templates, which we'll get to in a minute, where you fill them out and then submit during the window. The heavy lifting of the data submission is on the submitter, that's you in this instance. So this is where we're going to focus our time today. One major point here is that all data, regardless of how we receive it for the integrated report, will be made publicly available through our online database called Aquams. Our data requirements. Data received via each of the three data sources I just mentioned are subject to the same data quality and metadata requirements. We request that all data be collected under a project plan that includes QA and QC protocols for both collecting and analyzing samples. We also require widely approved sampling and analysis methods to be used so that data is comparable across assessment units of the same type. All data submitted during the call for data is expected to be post-processed, meaning that it has passed all QA, QC in the submitting agency's project plan. There are also certain requirements for data submitted during the call for data, such as being relevant to Oregon waters, to be collected within the data window, be related to a specific location, meaning we need to have coordinates, contain sufficient metadata, and if numeric and not narrative data, that it be submitted using the data templates that I'll show you in a few more slides. So quickly, some information about our submission window. This year, unlike last year, there will be just one call for data, meaning that all inland, estuarine, and marine data can be submitted during this call. The window opens on March 25th and closes on May 21st. That's a 60-day window, which we do not plan to extend so that we can keep up with our deadlines. Submission of data is completely voluntary. If you choose to submit data, though, we will assess surface water collected in lakes, streams, rivers, as well as marine data collected within 3 nautical miles of shore. For the integrated report, we do not assess groundwater, storm sewer, or wetland data. Okay, from here, we're going to move into our live demo. You can find all of the templates at this address, which is the same place that you access this video, so probably a bit repetitive, but it's better to be sure. All right, here we are. I have open our grab data submission template. For those of you who have used our templates before, this is going to look very, very similar. It's going to function in the exact same way that it has in the past. The major updates that we have made have been to domain values or our lists of valid values. So if you see up here, the last time we updated these was on March 10th, so very recently. And so for those, like I said, for those of you who have used our templates before, this is going to look and function exactly the same. For those of you who haven't, this will be a little bit more informative, and we will go through all of the different steps and what different items on our templates mean. We have, as you've seen on the website, I'm sure, we have this grab data template. We also have a continuous data template and a biological data template. I'll touch on the other two briefly after we go through this one. The main thing to know is that all of the color coding, all of the headings, information that I'll talk about now will be continued on and carried over into that continuous and biological data template. So all the functionality that you see happening here will also happen in those other templates. So we're just going to touch on those briefly. We're going to spend a majority of our time here on the grab template. So diving in, this is our instructions page. The first group of tabs you can see here is just for general information. You see down here it corresponds to the instructions and search tab, just as are listed here. We have the worksheet name and a short description of them. Moving on, the orange tab headers will give you information about the tabs. We'll go into these a little bit more in detail. But these orange ones are just information about what we'll need and what the different headers mean on the data entry worksheets or tabs, which are color-coded blue. And then finally, we have our green colored tabs that are lists of our valid values, or as I mentioned, or I called them at the beginning, our domain values. If you need assistance, we've got our email address or our website here. We also have our email address listed down here as well. So if you run into any issues, don't understand why something's not happening, if maybe I forgot to format a column so your dates look a little funny, that you can send an email to that, and I myself or someone else that receives the emails should be able to respond to you pretty quickly. We have briefly our column header guide over here. Yes, some of the colors are similar, but these ones refer to the column headers. These colors over here are the tabs. So if there's any confusion around that, again, you can always feel free to send us an email. One change on this page that may not affect very many of you, but if you are a new organization submitting data during a call for data for the very first time, you will need to fill out the organization details tab. We have moved that. Previously it had been one of the white tabs. Now it is the very first blue tab you see down here. None of the information that we're asking for has changed. We've just moved it closer to the data entry tabs rather than the general information tab. So no need to worry about this if you've submitted data to us for a previous integrated report call for data. If you're a volunteer monitoring organization or something like that, there's no need to fill this out. If it is your first time, though, submitting data to us, we do ask you to fill out these details, and we'll take a look at that in a second. What we're going to do first, though, is take a look at the Search tab. The Search tab has a bunch of information that should be helpful in filling out our forms. So if you have any questions around like how do I pick the right unit for my measurements of temperature or something like what's the right way, does this template want milligrams per liter with a capital L or a lowercase l, you can check that here by starting to type in milligrams per liter. Doesn't come up, you can say milligrams, sorry, and then click again, and we should have all of the different units that have milligrams in the name. We're looking for milligrams per liter, and it's up here. And this will show you that for this template, we want it to be a lowercase l. So yeah, if all of these will function the exact same way. You start to type something in, hit enter, and then you can click the down arrow and it should give you all of the available options for what you've typed in. We have a couple of different ways to search by analytical lab. We do ask that you use one of the labs from here. If you have not, please feel free to email us and let us know which lab you do use. For characteristic name, it's helpful to know if you need to include a sample fraction or if the speciation is required. Certain characteristics need that, so if we're doing phosphorus, we can click on that and we find out that no sample fraction is required and speciation is not required either. Let's see if there's another, you can also search by CAS number. If you put a CAS number in here and pull that up, you would see that this is for something that's very hard to say. So I won't try because I don't want to embarrass myself. But you can search by CAS number as well. And then also we have a search box for the analytical method. So if you're unsure about what analytical method, you can go through here, look for the characteristic that you've analyzed, and then it will show you what method ID to include and the method context to put on the results tab. Moving on, we're going to go to our information tabs now. Again, these just list out all of the column headers, sort of a maximum character length available, what type of data field it is, and then gives you a short description of what we're looking for in that column. Again, we have our column header guide over here, so you can see that these first four are required columns. The next column for the QUAP approval agency name is a conditional column, while the project attachment file name will be an optional column. You don't need to put the file name there if you don't want to. Conditional columns, when you enter data, will turn this sort of red speckled color if it's required based on a previous some other piece of information that you have entered, and we'll get to see that when we get to the data entry forms. But I want to just talk through it here because it's easier to see. Each of the info tabs will also have the number of projects or monitoring locations or results listed here. So as you can tell, if you have information for 50 different projects, which hasn't happened before, but If you did have information for 50 different projects that you wanted to include on just this one template, you could. We'll move over to monitoring location information now, and you'll see that that number now goes up to 250. Again, same table, giving the same general information. You see that we have all of our column names here, along with some that are required, some that are optional, or sorry, conditional, and some that are optional. Here we try to give a little bit more information about what we are expecting. For those of you that notice that some of these are underlined, that is because these are hyperlinks to information on our green tabs. So if you're not certain what type of monitoring location to call, if you have a monitoring location ID and a name, but you're not sure if it's a river stream or if it's an estuary, if it's a well is probably pretty obvious, but you can click on any of these hyperlinked column headings and it will take you to that list. So I'm going to click on this one, and now it has this kind of highlighted, I guess, in gray here. So you can see all of the different monitoring location types that are in the drop-down. The column headers are also hyperlinked in the data entry tabs. So you won't have to remember this as you don't have to remember all of these. There will be hyperlinked in the data entry tabs as well. Coming back here, let's see if there's anything else that we need to point out on this. I don't believe that there is. These are just for reference in case you have any questions. They answer some of the more general questions that you may have. We'll move on to the results tab now. Same table, same information, same column header guide. The number of allowed results is listed here, and it's to 10,000 results per grab template. So lots of space to enter your results in here. One thing that we do like to include here and to clarify is that field QC, such as duplicates and blanks, are accepted, but there's no need to submit laboratory QC, including blanks, spikes, or duplicates from the laboratory. That information we won't put into Aquums, but field QC, the duplicates and blanks we will accept. So just keep that in mind when you're including your data. We'll move on now to our first data entry tab. This one is not included. There's no project info on here because we included an example of a completed form over here on the right. So nothing new here, just a new location. All the other information that we're asking for is exactly the same. So again, so please feel free to ask questions if you have them, but I feel like this example should kind of guide you through what information we're looking for there. Moving on to the projects tab, you can see now that these are all the same headings that we already looked at in the info tab. You can feel free to put any project ID in here. It doesn't need to be numeric. It doesn't have to be all like letters. It can be letters and numerics. It can numbers. It can be anything. Project name is sort of like a longer description of it, and then the full project description can be, I think it was up to like, let's go back, almost 2,000 characters. So plenty of space. The most important part is that we want a unique ID for the project ID, and then sort of like a short, succinct name, and then the description can be the full purpose or summary. The approved QOP indicator is our first drop-down that we've encountered. You can see that this is just a yes or no choice. If you select yes, then the QOP approval agency, you can see now, is that speckled red color, meaning that we do need information there. So what we are looking for in this case would be whether or not DEQ has approved that, or if you have a different approval agencies such as EPA, Bureau of Recreation or Reclamation, sorry, not Recreation, um, any of those agencies. We just would like you to let us know that if you have a QUAP, who has approved it, and then you can attach it, submit that with your completed data template. We'll move on now to monitoring locations. Um, again, all of what we're looking for in each one of these columns is listed on the info tab, but we do include some boxes that help to kind of guide you along. So the text length for this is just kind of like letting you know that anything over 16 characters may be trimmed when it goes into Aquums, but you have up to 16 characters, and usually that's enough. We haven't really encountered any over 16 as of yet. The location name, that similar to the project name, this can be a little bit longer than the unique ID, but not the full description. We have that available down here in column I. Then we look for the monitoring location type. Generally, the information that we're receiving is river stream, but there may be other monitoring location types that you're submitting, like estuaries or lakes, things like that. Here we have a little note that the latitude should be 2 digits followed by, and then about 6 after the decimal. Longitude, it's important to include your negative sign just for where we're located here in Oregon. We have a drop-down list for the horizontal datum that the coordinates were collected in. If you are using Google Maps to collect your coordinates, that's WGS 84. Our coordinate collection method, again, if you're using Google Maps, that's interpolation or map. Unknown is also fine. And then if you're out and using a GPS, a handheld GPS, or some other instrument to collect your coordinates, then this would be the correct choice there. We have some additional conditional columns here. Again, whether or not your data was collected on tribal land is a drop-down, yes or no. If you select yes, then we do require you to include the name of the land where the data was collected. So that's important to fill out there. If you were to put something in here, let's see, then the red speckled goes away to show that you've fulfilled that requirement. And that continues on through here. We have a drop-down list for county. We have a drop-down list for state, but there's only one option, so don't get that one wrong, I'm sure. Let's see. If your monitoring location is also used by a different organization, you can put their information in here, so their monitoring location ID, and then include the context for that. So if it's a USGS site as well, you could put USGS in here. And then we offer 3 different spaces to include those different monitoring location IDs. Moving on to the results tab. Here we ask that you let us know which project the data that you are including on this tab is associated with. This really only comes into play if you're submitting data for multiple projects on one template. That's not usually the case, but it may be for you, so we make the option available. This dropdown will populate from the information that you include on the projects tab. So if you don't include any information on the projects tab because you've already submitted data for that project, and this is just a continuation on of that project, then this dropdown won't have anything and won't be populated by anything. But if we were to go back here to say, let's see, lake monitoring, and we go back to our results tab, you'll see that we can now select lake monitoring and then continue on with our data entry. We can also, obviously, if a sample was collected and it was going to be attributed to multiple projects, you have the option to put those here. This monitoring location dropdown is very similar. It's populated by the information that you provide on the monitoring locations tab. Otherwise, it won't have anything. For both the project and monitoring location tabs, if you have submitted data or submitted information about a project or submitted the monitoring location in the past, then there's no need to submit it to us again. That will save you some time in filling out this template. But if it's the first time that you have submitted data for the particular project or monitoring location, we do ask you to fill out information pertaining to both of those on the template that you're submitting the data or the results for. So if you have any questions about that, send us an email and we'll get that cleared up for you. The rest is somewhat self-explanatory. Activity start date, start time, end date, and end time. These will automatically update, but you can also fill them in. Some columns will be locked. I think we've got one right here. This column will be locked, meaning you cannot paste over it. So if you are saving time by taking your analytical results or results that you've received from your analytical lab and trying to just copy them and then paste them into this template, because you've got all of the columns are already lined up in the export you've received from your lab, you will get an error if you try to paste over this column because there's a protected formula in there. So our suggestion is to copy columns A through O, paste that in here, and then copy columns Q through the end. I forget what letter we go to. Let's just look real quick. Through AT and paste that in. That way you can still save yourself the time by copying and pasting, but you won't run into the error that you would get if you tried to paste over column P because it is locked. We have the columns in a specific order because we have import configurations that allow us to easily upload this information to our Aquams database. So we ask that you don't rearrange the columns in any manner. You shouldn't necessarily be able to because we lock these templates down pretty good. But if you find a way to do it, we just ask you to please don't rearrange the columns. Similarly, if you find that you wanted to rearrange data by copying some columns and then, I should say, cutting some columns and then pasting them up higher, that may affect some of the conditional formatting that is in here or the formulas that are embedded in some of the cells. So we do ask that you don't try not to cut and paste within the template. If you're going to do anything, copying and pasting should be just fine. Cutting and pasting does tend to cause some issues. I'll just briefly touch on the green tabs. Otherwise, I'll leave them for you to explore. But again, these are lists of our valid values. They're loosely associated with the different data entry tabs, minus the search tab, which has longer lists, but we'll take a quick look. Here's the information that is in the drop-downs for the projects tab. Pretty short and sweet there. Monitoring locations has a little bit more data, but this is where you would be able to come and look at all of the different requirements for the drop-downs on the monitoring locations tab. Results is much bigger because it gives some definitions for different things, such as the media name. So if you're unsure if your sample is sediment or soil, you could have a description here. Probably more necessary would be whether or not, or to decide which activity type you're using. One change that we have made this year was to move the most commonly used activity types to the top of this list, which is also reflected in the dropdown. So a lot of the data that we receive is field measurements or observations. Those are would be data that you collect on a field meter. So think temperature, dissolved oxygen, pH. If you're doing a replicate of any of those, you would use this second option here, which is a quality control field replicate, measurement, or observation. If the data that you're submitting was collected in a water bottle, then analyzed by a lab, that would qualify as a sample routine, meaning it was a grab sample sent to the lab to be analyzed. Duplicates of those would be a quality control sample field replicate. It's very similar to the other one, easy to confuse, but just know that if it says measurement or observation, that's a result or a data point that's collected in the field, not something that is analyzed in the lab. Quickly looking at the valid values for the search tab, you can see we've got our units of measurement, the HUC 8 codes, our big list of analytical labs, then the very big list of characteristics and analytical methods. So those are all here. If you need to search through them, you can. Typically, we advise people to use the search tab, but if it's easier for you to do to use the find and select through here, obviously you can do that as well. Might make it a little bit quicker for you. It's just up to your personal preference. That kind of brings us to the end of our grab sample template. So I'm going to move on to the continuous data template. We'll go through this one relatively quickly just because it does function and look almost exactly the same, which I hope you can see just by looking at this instructions page. Again, we have all of the different info tabs down here that are going to do the explaining of the different column headers and what we're looking for on them. A couple of differences here. Monitoring locations, projects, those are the same. We do have tabs for new equipment. Equipment is very similar to projects and monitoring locations in that if you've submitted data for a piece of equipment in the past, meaning we already have it in Aquams, there's no need to submit the equipment information to us again. Deployment information, you will need to submit deployment information for any logger that you are putting out that will have data included on the results tab. If we find kind of things aren't in balance, like if you've got results but no deployment information, then we'll contact you. But you see here the type of information we're looking for. What piece of equipment was it for? What monitoring location was it collected at? The activity start time and end time for this deployment. What type of media was it in? What project should it be associated with, etc. Then we can move on to the results. This one's pretty straightforward as well. A lot of required columns in here. This one, there aren't any locked columns, so this makes it a lot easier to copy and paste. For your convenience, we do have a fairly lengthy or a big number of results that you can paste into here. It's 1 million rows. That does mean that the file size itself is going to be very large. We suggest when submitting large continuous files like that you try to zip them, and then you probably have to email them to us rather than use the data submission form. We ask for audit data. If you're putting loggers out, we're certain that you're doing data audits to ensure that there hasn't been drift, things like that in the data that you've collected. So here's that audit tab. We have put a little note here about which equipment ID to put here. We want the ID of the logger being audited, not the equipment ID of the equipment performing the audit. So this should correspond to the logger that's been in the water, that's staying in the water, not the meter that you're using to test the temperature alongside the monitor or the logger while it's been deployed. Let's see, anything else to point out here? The valid values tabs are almost exactly the same. So they're going to function the same. if you need to understand or get an explanation about what those activity types are, here they are again. Um, result value types, trying to understand like what's a calculated value versus an actual value, things like that. Search tab, a little bit shorter here, but it's got analytical methods and characteristics. Then the units of measure and Huck8s are all the same there. Just need to touch briefly on the biological template, but again, we can breeze through this. It all looks the same. It all functions the same. White tabs are for general information. Orange tabs are for info on the specific data entry tabs. And then we'll get into our actual blue data entry tabs here. And then we have one tab for valid values. Projects, monitoring locations, those are identical to the other two templates. Samples are slightly different. For this one, we do ask that you give us a habitat type. And I will make sure that these drop downs are working when we have these ready for you. They should be working now. But that's okay because the options will be there when you go to use the templates. That's why we do live demos, right? To embarrass the people that created the thing when something doesn't work. Yeah, a lot of this information is the same. This lab number would correspond to the information that your taxonomic lab attributes to the sample that you've provided, just to help keep that straight for you and for us. So that's just the sample, and then the results in here. The sample ID will come from this column, which is generated. As you can see, it's locked and has this crazy formula in there. That will be available in a drop down here once you've filled out your sample information. The DEQ taxon code is something that you can find either on the search tab here. You can search by common name or by scientific name. If it's easier for you, you can also do you just got the taxon here. You can do find and select on here to try and find that. The DEQ taxon code is the value in this Q column, and that's what we want to have here. When you fill out this column, these two columns will automatically populate. If there is no DEQ taxon code for the taxa that you have results for, we ask that you provide us the lab ID taxon, so your taxonomic lab, whatever they tell you the taxon is, we ask that you include that here, and then fill out these conditional columns so that we can update our list and to accurately reflect the data that we're receiving. Stage ID, this is our short list. You have adult, larva, pupa, and unknown. This is your result, so the individuals that you counted. And then for unique taxon, whether or not the taxon itself is unique within that sample. For adults and larva in the same sample, this would obviously be no. If it is a unique taxon, meaning you only found it once in the sample, that would be yes. Any comments that could help us differentiate between two seemingly similar results? Say you had 15 larvae of one taxa, but then you had 3 that were close but were damaged, or if you had, and so you couldn't necessarily say that they were the exact same taxa, whether that's to genus or to species, then it would be really helpful to have those comments in here. Just for us, for our purposes, we do audits of aquams, and sometimes these comments can be super helpful. I believe that's it. Like I already kind of showed the valid values tab here. So I think that kind of brings us to the end of our live demo, and I'll leave it at that. One data type that our templates don't cover is non-numeric data. This type of data cannot be tabulated in a spreadsheet. For instance, journal articles, state reports, or findings from cleanup efforts as shown in the pictures on the right. A couple of requirements if you plan to submit non-narrative data. The data must be related to a specific location so that we can attribute the data to an assessment unit. Using the cleanup effort example, this would be the coordinates of the starting and ending location of the cleanup event. Your data submission must also make reference to the beneficial use that is being impaired. So going back to our cleanup effort, the beneficial use that's being impaired here would be the aesthetic and recreational use, beneficial use. Narrative data can be submitted using the same process as numeric data, which I'll describe next. So when submitting data, you can go to the same web page where you found this video. Again, the dates that we're accepting data are from March 25th through May 21st, 2025. The data window itself that we're looking for is January 1st, 2020 through December 31st, 2024. At this website, you will find a short online form with information that we need, including contact information, the name of your submitting agency, and how you verified your data. There are two ways to submit data. For files under 20 megabytes, you can attach the files directly to the online form before hitting submit. For files over 20 megabytes, these files will need to be emailed to datahelp.ir@deq.oregon.gov. That does bring us to the end of our tutorial video here. If you have any questions about the template or how you need to submit the data, you can send those questions to datahelp.ir@deq.oregon.gov. And if you have any questions on methodology or have any comments, you can send those to integratedreport@deq.oregon.gov. Thank you very much for watching this video. Best of luck with the templates, and please reach out if you have any questions. Thank you very much.